Membrane proteins are key targets for many drugs. They are located between the outside and inside of our cells. Some of them, called transporters, move certain substances in and out of the cellular environment. Yet, extracting and storing them for observation is particularly complex. A team from the University of Geneva, UNIGE, in collaboration with the University of Zurich, UZH, has developed an innovative method to study their structure in their native environment, the cell. The technique is based on electron spin resonance spectroscopy. These results, just published in the journal Science Advances, may facilitate future development of new drugs. In living organisms, each cell is surrounded by a cell membrane, or cytoplasmic membrane. This membrane consists of a double layer of lipids. It separates the contents of the cell from its direct environment and regulates the substances that can enter or leave the cell. The proteins attached to this membrane are called membrane proteins. Located at the interface between the outside and inside of the cell, they carry various substances across the membrane, into or out of the cell, and play a crucial role in cell signaling, i.e. in the communication system of cells that allows them to coordinate their metabolic processes, development and organization. As a result, membrane proteins represent more than 60% of current drug targets. Difficult objects to study. The biophysical study of their structure, the spatial organization of the constituent amino acids, is therefore essential. To characterize them, scientists must extract these proteins from the cell membrane in which they are found and isolate them from all other proteins. Once extracted, membrane proteins cannot be studied in aqueous solutions. They must be maintained in liquid solutions composed of detergents. They can also be inserted into artificial membranes called nanodiscs, made of proteins and lipids, or in pure lipidic membranes. In any case, these strategies remove them from their physiological environment and do not allow their functioning to be finely observed in situ. Proteins outside their native environment might show different structural properties, therefore misleading drug development. A revolutionary method. A team led by Enrique Bourdignon, full professor in the Department of Physical Chemistry at the UNIGE Faculty of Science, in collaboration with Marcus A. Seeger, associate professor at the Institute for Medical Microbiology at the UZH, has developed a new method for studying MEM. To achieve this, the research team relied on a specific tool, nanobodies. These are fragments of antibodies that are able to recognize and bind to a specific target, such as an antigen or, in our case, a membrane transporter, in a very efficient way, explains Enrique Bourdignon. The scientists have thus artificially produced specific nanobodies for a membrane transporter and use them to directly report on its structure. Inserted into E. coli cells, 2 nanobodies target the desired membrane protein on the inner membrane of the cell and attach to it, explains Marcus A. Seeger. The multidisciplinary team also included scientists from the Ruhr University Bochum, Cluster of Excellence RESOLV, and the University of Osnabrück, Germany, and the University of Southampton, UK. New targets for certain drugs beforehand, a small magnetic probe, a molecule carrying unpaired electrons, was attached to each nanobody. When 2 nanobodies bind to the transporter, we can measure the distance between the 2 magnetic probes in cells using our EPR methods, explains Enrique Bourdignon. This technique is called electron paramagnetic resonance spectroscopy, EPR, or electron spin resonance. The distance measured is in the nanometer range, 1/1,000,000th of a millimeter. For the first time, we have managed to obtain a clear picture of the conformation of a membrane protein in its real environment, and we could follow the change induced when we modified one single amino acid into another one, enthuses Enrica Berdignon. The development of this new strategy is the result of excellent and challenging teamwork between our two groups at UNIGE and UZH. In particular, it is the resilience of the two first authors, Dr. Laura Galazzo, UNIGE, and Dr. Gianmarco Meyer, UZH, that made this project a success after 5 years of research, underlines the researcher. This new strategy allows a precise determination of membrane proteins' properties in their direct environment. It offers the possibility of better understanding how these proteins transport certain substances into and out of the cell. This method also has the advantage of being easily transposable to mammalian cells. It could then be used to better understand and therefore better target the membrane proteins which reject certain anti-cancer drugs outside the cell, and thus combat the phenomenon of multi-drug resistance. Make a difference. Sponsored opportunity story source. Materials provided by Université de Genève. Note: content may be edited for style and length. Journal reference: Laura Galazzo, Gianmarco Meyer, Doval Janaline, Christian Perry, Dario De Vecchis, Bianca Stridnig, Hubert Hilba, Lars V. Schaefer, Ilya Kuprov, Arne Moeller, Enrica Berdignon, Marcus A. Seeger. The ABC transporter MSBA adopts the wide inward open conformation in E. coli cells. Science Advances, 2022. 841. doi. 10. 1126, Sci-Adv. ABN 6845. Cite this page, Université de Genève. A revolutionary method to observe cell transport. Science Daily. Science Daily, 24 October 2022. ift.tt/fr8sinw. Université de Genève. 2022, October 24. A revolutionary method to observe cell transport. Science Daily. Retrieved October 24, 2022 from ift.tt/fr8sinw. Université de Genève. A revolutionary method to observe cell transport. Science Daily. https://ift.tt/fr8sinw. Accessed October 24, 2022. Thanks for watching. Please subscribe my channel for more videos.